But in the meantime, as I mentioned, I'll be briefly giving your background information to our attendees. So Dr. Lingyan Shi is currently assistant professor in the Department of Bioengineering at the UC San Diego in the engineering school. And she received her PhD in biomedical engineering from the City College of New York, CCNY. She was a postdoc at the Institute for Ultrafast Spectroscopy and Lasers at CCNY. She then moved to the Department of Chemistry at Columbia University for her second postdoctoral training in biophotonics prior to her joining UC San Diego. And Dr. Shi major scientific contributions include the discovery of the golden optical window for deep brain imaging and microscopy for optical imaging of metabolic dynamics in living organisms in situ. And Dr. Shi also won multiple awards, including the Blavatnik Regional Awards for Young Science in 2018, and many others hope to come soon. And her primary research interests focus on developing and applying novel optical techniques for studying neuroscience, cancer, and metabolism. And without further ado, I think we are happy to have Dr. Shi today. And please, you may want to start sharing your screen.
Thank you so much, Ahmet. I would like to share my screen first. Thank you for the great introduction and for the kind invitation. So I'm very happy to be here today to talk about visualizing metabolic activities in cells and animals with vibrational imaging. So before starting the technology part, I would like to say something about metabolism. Metabolism is a process that occurs within a living organism in order to maintain life. So it's very important. The molecules that are involved in metabolism, including carbohydrates, proteins, lipids, DNA, RNA, and other molecules that you can name all. For example, lipids metabolism. We are very familiar about lipids. So we know that the food provide nutrients, and we can store them, convert those nutrients into fat and store in the liver. When the organs such as brain or muscle need it, they will convert it back into glucose and let the organ use it. So along the human evolution, our body has accumulated more and more fat compared, in the past. Why? Because of the lifestyle change and also the diet change, we accumulate too much fat that actually it induces diseases, even cancer. So in the hospital, when the medical doctor wants to localize where is a tumor in the body, they would like to use an imaging technique called PET. Before image that, they will inject a glucose analog. Because the tumor cell have a higher metabolic activity, they tend to absorb or uptake more glucose analog into their tumor cell. That's why we can visualize those tumor locations. But those glucose analog cannot be metabolized. They just allow the cell to uptake them. So it's just uptake, not metabolism. And the imaging tool that we visualize metabolism, including PET, MRI, fluorescence, we are very familiar of, and they have different physics principles, like radioactivity within nucleus, the spin of nucleus, and the fluorescence microscopy is the electron outside of the nucleus. So what about the motion between nucleus? We call vibrational spectroscopy. So all the devices we have seen in the hospital or in the lab, the light microscopy here, actually this microscopy, we can integrate our vibrational spectroscopy like a stimulated Raman or other Raman technique into this fluorescence microscopy and visualize those chemical bond vibration. So this is a typical PET image of the brain. This is MRI. Those images provide the metabolic activities from the body. However, they do not provide cellular resolution or subcellular resolution. So for the biologist and the scientist, medical researchers, they would like to see cellular or subcellular resolution of the metabolic activities. So they tend to use the optical imaging techniques such as fluorescence and Raman microscopy. So for the fluorescence microscopy, it's very popular and widely used, not only because it provides high resolution, but also because there are a bunch of commercial available fluorophore or probes that we could purchase them and easily target those molecules of interest that we would like to see. In contrast, for Raman microscopy, we know that Raman spectroscopy tells the Raman spectrum tells the chemical bond information. So in theory, we could just pick one of the peak and visualize the chemical bond to see the molecule's distribution. However, in reality, it's a little bit difficult because the spontaneous Raman have a limitation of a sensitivity. So we have to take a long time to acquire an image. And it's also easily overwhelmed by autofluorescence. So in the past decades, people are working on how to enhance the sensitivity. Not until 10 or 11 years ago, when people in Harvard University, including Dr. Sunny Xie and Ming Wei, they invented a new type of microscopy called stimulated Raman scattering microscopy. So it combines the Raman scattering and stimulation emission together. So this, instead of using only one laser like spontaneous Raman microscopy, the stimulated Raman microscopy used two laser beams, the pump laser beam and the Stokes laser beam. So when the energy differences between two laser beams matches the vibrational modes of the molecule of interest, the molecule starts to vibrate dramatically enhance the vibrational rate, and the signal, the sensitivity can be enhanced up to 10 to the 8 magnitude. With this kind of sensitivity, we can easily get video rate imaging for the chemical bond in the molecule. So in the lab, we build our own microscopy with two laser beam in and then a modulator on the Stokes beam. And we put a sample here on the object side and collect those with a photodiode. Of course, we have to use a block amplifier to have the signal better. So the key trick here is the modulation, because we are detecting the laser beam intensity changes. Compared to the laser beam itself, the intensity changes is relatively small. So we want to remove all the noise from the laser side and achieve the high sensitivity. So after we build this microscopy, we are happy to take those label-free imaging from mice or zebrafish or other cells. So this is an example of a mouse skin tissue. So you can see the bright shining stuff. This is the sebaceous glands in the skin. We can see there are dark spots in the middle. That's the nucleus, because we are detecting CH2 for the lipid signal. So if we tune the laser to pick up the signal from the nucleus will be bright, and other parts will become darker. This is a lipid signal, and this is a lipid signal from a zebrafish. So we take very nice label-free image. However, we know that the fluorescence microscopy can see a molecule of interest that you really want to see specific proteins. So we are not satisfied with the label-free vibrational image. We want to do labeled vibrational image. And labeled vibrational image allow us to see something that the fluorescence microscopy cannot see. So what are the fluorescence microscopy cannot see? If we recall those fluorescence probes, they are actually relatively big compared to some molecules. So this is an example of an antibody. This is quantum dots, 5 nanometer diameter. Fluorescence protein is smaller, but even organic fluorophore, they are smaller than those probes, but they are still bigger than glucose or amino acids. So imagine if we want to visualize those small metabolites like glucose, amino acids, water, fatty acids, choline, acetate. If we label them with a fluorescence probe, it will be too big. And this big fluorescence probe will perturbate the function of those small metabolites. So now the idea is we want to tag those small metabolites with a very little tiny tag, and this tag can preserve the Raman vibrational signal, even those small metabolites incorporated into other bigger macromolecule, this small vibrational tags information is still preserved, and the function of those small metabolites will not be perturbated by this small tag. The idea is we want to see the signal and pick up the signal among these Raman spectrum. If we look at the Raman spectrum from a typical biological sample, you will see 3 different regions. This is a fingerprint region, cell silent region, and the CH stretching region. We already noticed that their cell silent region does not have any signal from 1800 centimeter inversed to 2800 centimeter inversed. So the idea is we are going to utilize this cell silent region because all the biological sample naturally does not have any signal in this region. So if we tag this small tag with a vibrational modes in this cell silent region, then we can pick up the signal with no background. So these are fingerprint region signal from the endogenous chemical bond in the biological sample. This is also endogenous. So this one can separate the signal from those endogenous chemical bonds, so we can visualize them clearly and see the metabolic activities in the cells and tissue. First experiment was done by Fujita's group in Japan. They used a tag called Alkyne, the carbon-carbon triple bond. So they attach them into EDU, and when the cell synthesizes new DNA, they will see here the EDU signal is separated from other endogenous chemical bond for the cell. So we can see which cell is dividing by just the look at the EDU signal, because only the cell is dividing, they will have this newly synthesized DNA. With this in mind, we can visualize the DNA distribution when the cell is dividing. If you take an image in time lapse, so in time theory, you can see from the time 0 to 30 minutes, how the DNA distribution inside a single cell. And also from a piece of tissue, if you culture them with Alkyne labeled EDU, then you will see which cell is dividing. This is a piece of a brain tissue. So if you look at this lipid channel, you will see how the lipid distribution inside this piece of brain tissue. There are a lot of voids here. They are the nuclears without the lipid signal. But if you tune the laser to visualize the Alkyne, the carbon-carbon triple bond, you will see this cell and this cell, they are dividing, but not other cells. So you can visualize those cell metabolic activities. And we can also label Alkyne bonds onto other molecules to see, for example, the phospholipid synthesis with choline and other molecules for protein synthesis, for triglycerides, DNA, RNA. We could utilize this technique and expand to other kinds of molecules. However, those molecules is not perfect, if we want to do in vivo experiments on animals. So there is another kind of molecule that we can attach, the atom that we can attach to the molecule is called a stable isotope. So those stable isotope, including deuterium, are very ideal for utilize them as a small tag onto the small metabolites. So my favorite is deuterium atom. It was discovered by Harold Urey, who won Nobel Prize in chemistry in 1934. Compared with the hydrogen atom, the deuterium has an extra neutron, which makes this atom heavier. So what will happen if this deuterium atom incorporated into a biological tissue, even an animal? So let's see this spectrum again. If the CD bond formed in the newly synthesized macromolecule, there is a new chemical bond called carbon deuterium. So the carbon deuterium bond vibrates inside the cell silent region, which does not have interference with other signals in in the endogenous chemical bond. So we can easily visualize the metabolic activities by just adding the deuterium molecules, deuterium labeled molecules. Then we can expand the application to see more in vivo or in the tissue level isotope labeled metabolic activities, such as protein, lipid synthesis, DNA, RNA, carbohydrates. So we call this kind of imaging technique bioorthogonal chemical imaging, and I have separated them into two different categories. The one is called carbon source probe. This category, we have those small metabolites. They are already nutrients like amino acids, palmitic acids. These are fatty acids or acetate glucose. Themselves are already nutrients. And another category is non-carbon source, like heavy water, drinking water. These just water, but they participate in metabolic activities. They do not provide carbon themselves. For the carbon source probe and the non-carbon source probe, I would like to introduce in the next few slides separately, and how different they are. So before that, I want to say a little bit about why we want to visualize metabolic activities in situ. Because direct visualization of metabolic activities in living organisms is very important to understanding many biological processes. And it is the metabolism, such as protein, lipids, that carries out the genetic blueprint, which cannot be imaged in vivo by using traditional methods like PET, MRI, mass spectrometry. And we just developed this technique to probe the metabolic activities in biological systems, including small animals, or even we could use it in human to do some clinical study and visualize metabolic heterogeneity to identify where is the newly formed myelin, where is the tumor boundary, or we do study on postnatal development or aging process. Now, I would like to give you some examples that how we did this. For example, the first image I show here is a cell that uptake the amino acids. So we put deuterium labeled amino acids in the cell culture medium. Then the cell will use them to synthesize new protein here. So the new protein have a deuterium atom, which is the CD bond that we want to collect with the Raman imaging. So now this peak is in the cell cycle region. So we select this peak and image this chemical bond. We can see where is a newly synthesized protein in the cell. And those are the distribution of the newly synthesized protein. And those are the neuron cells. We know that neuron, when there's a piece of memory formed, there must be a new piece of protein synthesized. So we can visualize where is a new synthesized protein located in the neuron. And we can enlarge those dendritic spine to see how the protein synthesized in the dendritic spine. And we can also inhibit those protein synthesis if we put some inhibitor. We can see that the neuron with inhibitor, we cannot see the new protein synthesis anymore. And for protein synthesis, we can do time-lapse image to visualize how the cell synthesizes a new protein with a time course. So we can even see the dividing cell, how the newly synthesized protein distribution change over time. And we can also use a specific amino acids, for example, glutamine. So use this glutamine to visualize the tumor cell metabolism. For example, the tumor have a different metabolic pathway when they are under stress, under hypoxia. So this image shows that under normoxia and hypoxia, they have a totally different metabolic activity with the deuterated glutamine. And I introduced a lot of cell work. Now I would like to show you some in vivo work because you would like to see how we do it in the animal. So for the deuterium labeled amino acids, we have to do a little surgery to inject those amino acids, deuterium labeled amino acids. So we can pull out the carotid artery and cannulate with a catheter, actually implant the catheter here, and we connect it with a syringe. The syringe pump can provide a constant flow rate to pump in the deuterated amino acids. So the amino acids will be directly pumped into the carotid artery and bypass all other organs and directly reach the brain. So the brain can use the amino acids to synthesize new protein. So we can visualize how the brain synthesizes the new protein in different brain regions. Here is an example of a hippocampus. So we can see that the CD region here, if you tune the laser to visualize only the CD labeled protein, we can see the new protein, how they distribute in the hippocampus. And this is endogenous CH label-free protein. This is endogenous label-free CH2 lipids. We can see how different they are. And the hippocampus seems to have a lot of new protein synthesized because it's a very important region for memory. So compared with hippocampus, the cortex has a totally different phenomena here. If you visualize, if you see those small dots, the green dots here, they are the cells that actively synthesize protein. So if you compare with the label-free protein channel, those cells are very active, but other cells are not as active. If we merge the two channels together, the yellow dots shows the only active cells in the cortex that synthesize new protein. So it's already tell us different regions have different metabolic activities to protein synthesis. And the brain have another region which is very interesting. For example, the choroid plexus. As an adult like me, I produce 500 milliliter CSF, cerebral spinal fluids, to maintain the brain health. So in the mouse, this piece of tissue also is a region, the tissue to produce the CSF. So the choroid plexus located inside the brain ventricle. It's like a floating seaweed, and it is producing the CSF here to maintain the brain pressure. And you can see the new CD protein channel. These are the protein signal that newly synthesized by the choroid plexus organ. And compared with this endogenous label-free channel, we can see here the brain tissue. These are the two pieces of brain tissue, and they are not synthesized so much protein as the choroid plexus did. So we can also see different time length, the different duration. So but 8.5 hours compared with 2.5 days, the protein synthesis amount is different. And we could quantify them by divide with this image by this original protein image to see the protein turnover rate, like how much % of the protein turn over in this region. So we can see it's about 6% will turn over, but shorter time have a less turnover rate here. All right, so I talked a lot about protein, and I think you also are curious about fatty acids. Now I want to introduce something about imaging the fatty acids metabolism. For fatty acids, people are concerned about the saturated and non-saturated fatty acids. If we look at the chemical bond, they have differences. The saturated fatty acids does not have a double bond, but the unsaturated fatty acids have a carbon-carbon double bonds. So how different they are metabolized in the cell? Bear this curiosity, we did some experiment on single cell. This is a single cell. If you see the voids inside in the middle, this is nucleus. So we only take the image from the CD labeled lipids, so we can see this lipid distribution. With saturated fatty acids in the cell culture medium, the cell synthesizes those CD labeled lipids on the ER membrane, and the ER membrane becomes solid. So the phase from the fluidic phase becomes solid phase. Actually the saturated fatty acids has a toxicity on this cell membrane, but unsaturated fatty acid does not have that phenomenon. We put the same amount, but the cell only only store those unsaturated fatty acids into those lipid droplets as an energy source, but the membrane is still fluidic and they are healthy. So they are completely different phenomenon. And if we see this cell cultured with a saturated fatty acid and become solid membrane, the ER membrane becomes solid. If we put some unsaturated fatty acids, it can be rescued. So the membranes start to be dissolved and return to fluidic status. So that is a very good phenomenon that we can see that how we should eat our meals. So remember, if you want to eat junk food, those have a lot of saturated fatty acids. So we should add those unsaturated fatty acids from olive oil, fish, and nuts to dissolve those solid membrane. And now I would like to talk about a very interesting application, because we know we can visualize those metabolic activities and how we could see the heterogeneity in the solid tumor environment, because tumor have many different kinds of cells mixed together, not only tumor cells but also like macrophage, adipocytes, cancer stem cell, T cells, or blood vessels. So it's a bunch of a mixture inside a solid tumor, and every type of a cell have their own metabolic activities. So it's very important to see how the tumor grow with metabolic heterogeneity. So before doing that experiment, first we did a xenograft to grow a tumor inside the mouse brain. And when the tumor grow into when the tumor cell grow into a solid tumor, we inject the palmitated acids with the same method we introduced here before. So this is the palmitated acid that introduced into the carotid artery. So this mouse is still walking around and doing the normal daily activity, but the brain is used this deuterium-labeled palmitated acids to synthesize new lipids. So this image is a cross-sectional area of a mouse brain that contain a glioblastoma tumor here. We can see it from label-free SIS image by two-channel image. So the green channel is the lipids channel, and the blue channel is the protein channel. We can already see the tumor location here, because the brain tissue is very special. It has a lot of lipids. So when you take a lipids channel, you have a lot of green color here, but the tumor does not have that much lipids. The glioblastoma have a lot of protein instead. That's why you can see the blue color is very intense in the tumor, but the green color is very intense in the brain tissue. However, we cannot see how the metabolic activity occur in the tumor. So we use the label technique to see how the tumor metabolize the palmitated acids. From this image, we enlarge the tumor here and look at the different region of the tumor. You can see that the tumor boundary on the edge of the tumor have a higher metabolic activity of palmitated acids, not in the middle or central brain tumor. So we can visualize how heterogeneous it is. You know, the tumor synthesize those palmitated acids on the edge, on the boundary, or those cells are tended to invade into other regions of the brain. So they are more actively dividing. And now maybe you want to ask if we can visualize the protein and the lipids metabolic activity together with two-color image. So I did a small experiment to prove if we put amino acids only, we can see amino acids synthesized as a new protein here. If we put palmitated acids only, we can see palmitated acids synthesized lipids here. If we don't put anything, we don't see any new macromolecule here. So if we put both, we can see both. We can see the protein here, the lipids here. If we merge them together, we can see two-color image in the neuron. So we can do two-color image. Now I want to introduce another one just briefly. It's acetate. So we can also visualize the acetate incorporated into lipids in the cell. So under the normoxia and hypoxia, the acetate also have a different metabolic metabolic pathway to do the incorporation into the lipids or ER membrane. And it's very interesting to visualize that in the single cell level. Now, I would like to introduce another platform. I separate the glucose from all other carbon source because glucose is so important and very unique molecule. And I want to talk about glucose metabolism as a single platform. So this video you already see in the beginning that I show PET image, they visualize the glucose uptake. Those glucose analog they use cannot be metabolized in the cell. And this is another image with Alkyne labeled glucose. So these glucose also can be uptaken by cell, and we can do SRS image on those uptaken. However, if we fix the cell with paraformaldehyde, those signal will be all gone because they are not synthesized they are not incorporated into macromolecule. They are just being uptaken inside. So they cannot see the macromolecule and incorporation, which means we have to image them when the cell are alive because they can wrap those small molecules inside. So the difference is if we use deuterated glucose as a carbon source metabolite to probe the metabolic activity, these deuterated glucose are truly metabolized by the body, and the body will synthesize them into glycogen, into DNA, RNA, into lipids, protein, or they can be simply utilized as an energy source and generate ATP. So now the question is, can we just visualize those different macromolecules? Because we already see 5 different macromolecules. Can we visualize them simultaneously and separate those signals? Just only one glucose labeling, but you see 5 different molecules. So bear this question in mind. We did a simple experiment in cell culture first. Let the cell incorporate those deuterated glucose. Then we extract the lipids, protein, DNA, RNA, and the glycogen that is synthesized inside cell, purify them, and measure the Raman spectrum of each macromolecule. So from the Raman spectrum, we can see every molecule can be separated by the Raman spectrum, which is very good. That means we can also separate them in the image. So we did the image here on the single dividing cell. And when we take image, we just pick those protein peak, lipids peak, and DNA peak with 3 different channels, and use this algorithm that derived from the Raman spectrum here. Then we can mix the signal for purified DNA and the lipids and the protein. So you have a 3-color image of a single dividing cell. So these are the newly synthesized DNA, lipids, and protein inside of this single cell. And also we can do it in the animal. If we let the mouse drink the glucose water and use the same method. For example, we take out a piece of liver, and the liver has newly synthesized macromolecule inside. So we use this algorithm to mix the signal and derive the 3-channel image of newly synthesized lipids, glycogen, and protein. Of course, we can also have other like label-free image for lipids, glycogen, and protein, and calculate the turnover rate of each macromolecule that is derived from the glucose. Now we can expand this method to visualize different organs' metabolic activity like heart, pancreas, kidney, small intestine, stomach, and the fat pad. You already noticed that the fat pad does not have a lot of deuterium-labeled protein synthesized, but a lot of lipids, newly synthesized lipids, CD-labeled. However, other organs like heart, pancreas, kidney, those have a lot of CD-labeled protein. So those protein cells those protein signals are derived from the deuterated glucose. With this method in mind, we can actually apply to solve some real biological questions. It's interesting to visualize those myelin. So myelin is important that it's wrapping outside the axon to protect the axon for signal propagation. But if this myelin sheath is damaged, a lot of neurodegenerative disease occur. So questions like how are myelin sheaths formed in child, how are myelin sheaths renewed and remodeled in adults, they are quite important. So we did an experiment on mice. We let the mouse mother to drink glucose. The mouse mother just had a bunch of pups, and it will feed those pups with milk. The milk have deuterated glucose. So after a few days, we look at the brain of those pups, and also look at the brain from this mouse mother and compare the signal. We can see the pup have a lot of CD labeled newly synthesized lipids in the brain, and the distribution is very interesting. You can see different time window have a very different lipids distribution. But in the mouse mother's brain, we did not see that kind of phenomenon. This is a label-free channel. We can see the mouse mother also have a lot of lipid signal, but not lipid metabolic activity. Not that much. There are a little, but not that much. However, the pup have a lot of new lipid synthesis. So we can see how they do the myelination in different time stage. Actually, the second week is the most intense myelination time window for pups, and also other parts like cerebellum, and we can also do quantification, how much of the lipid synthesized in each region, and compare, do the quantification to compare with the adults. Adults does not have that much, and the distribution of the lipids is very interesting here. We can study how they turn on those lipids synthesis at a certain time window. And another application we did is for small intestine, like the pup absorb a lot of lipid droplets from the milk, and they started to absorb by the small intestine cells, and the cell absorb them from the top. This top means towards the milk, towards the lumen of the intestine, but this bottom is towards the lymphatic system. So from top to bottom, we can see the lipid droplets become bigger and bigger. If we focus from the top to bottom layer, like this, the 0 micron, 7 micron, 14 micron, we can see the lipid droplets become bigger and bigger. So we can see the spatial distribution of different size of the lipid droplets in the small intestine cell. This is the first time we can visualize that kind of phenomenon. It means the small intestine cells are repackaging those absorbed lipid droplets, and eventually make a bigger, larger lipid droplets to let the lymphatic system get them into the circulation. It's a very interesting topic, actually. So I also want to introduce another very important imaging technique called pulse chase, because we know that the glucose have 7 hydrogen, so we can let the 7 hydrogen all replaced by deuterium, or we select 2 of the hydrogen atom and replace with only 2 deuterium. So D7 and D2 glucose, they have different Raman spectrum. The question is, if we use them to derive new lipids, the D7 derived lipids compared with D2 derived lipids, do they have different Raman signal? The answer is yes. So we can utilize this information to do pulse chase. For example, we give the animal 24 hours of D7 glucose, then stop giving D7 glucose, but only give D2 glucose. So we can visualize the sebaceous glands in the skin, how they turn over, how they derive the lipids. D7 derive the lipids distribution is the red color D2 glucose derived lipids is a green color. If we merge them together, we can see the outer layer, all the outside of the subsites, they synthesize the lipids more rapidly. So they have a faster lipid turnover compared with the lipids, the subsites in the center of the glands. So that's why we can see the two color pulse chase image with two different types of glucose. So I will quickly also go over heavy water platform. This is dual SRS platform, and also it's non-carbon source. Non-carbon source, it doesn't perturbate any metabolic activity. It's just the water. So the animal drink water here, and they will participate in the metabolic activity and incorporate those deuterium atoms into macromolecules such as protein, lipids, DNA, RNA, and the carbohydrates. So if you look at here, you can see the deuterium atom incorporate in a sparse way. It's not like every single hydrogen can be replaced. You just plug in certain position. So that's also influence the Raman vibrational modes, the position of the incorporation. So they can fastly incorporate into small metabolites, then slowly incorporate into larger molecules like lipids, protein, DNA, RNA, and carbohydrates. Now we can visualize those cell image. So this is an example of cell culture that if we give heavy water to the cell, for 5 minutes, the cell does not have enough time to do metabolism. If we culture them for 24 hours, we can see very bright CD labeled molecules information here. So if we choose laser to visualize heavy water itself, the heavy water have an OD bond. That's what we want to see. If the CD bond information, the CD signal will be interfered with the OD signal. The answer is no. We can see if the cell have metabolic activity, it will form a new CD bond, but the CD bond have a different Raman signal from the original OD bond from heavy water. So we don't need to wash out the heavy water. Just let the heavy water be there. You just tune the laser to visualize the CD bond. And another point is, if the cell have no metabolic activity, we cannot see any new CD bond formed. So that's very, very good, because only if the cell is alive and doing metabolic activity, we can see the newly synthesized CD bond. And you also would like to see if we can do in vivo imaging. So this is the in vivo imaging example, how the mouse is synthesizing the new lipids in the sebaceous glands here. So we can see the blood flow here with the red blood cell floating, which means the mouse is still alive. And if we took a long time, we can see how they synthesize new lipids, how they deposit the new lipids. This is an example of a dancing C. elegans. So if we take a long time enough, we can see how the C. elegans synthesize new lipid droplets in the body. Now the question is, we know the heavy water will be incorporated into different macromolecules. The same thing, how we can visualize them simultaneously and separate the Raman signal. So we did a similar thing like what we did for the glucose experiments to extract those lipids, protein, and DNA, RNA, and we can visualize the Raman spectrum here and derive the 3 components algorithm, then apply onto the image to unmix the signal. Now we derived a single dividing cell have new lipids and a new protein and a new DNA signal derived from heavy water. It's very good that we can apply to different organs like adipose, axon, sebaceous gland, muscle, brain cortex, liver, pancreas, and so on. And you already noticed these 3 types of tissue does not have much CD protein, but these 4 types of tissue have a lot of new protein synthesis also. So it has a huge tissue heterogeneity in the body. With this in mind, we can apply this technique to solve some interesting biological questions. For example, the brown fat and the white fat in the obese mouse, how those fat metabolize. So for this experiment, we see the white type and obese mice have quite different metabolic activities. The heavy water will be incorporated into the newly synthesized lipids, and we can see more newly synthesized lipids in the white fat cells, not the brown fat cell. I put brown fat cell here. If you see the label-free channel, you can see the brown fat cell is here. The morphology is different. It has a lot of small lipid droplets inside, but the white fat cell have a very fat, bigger size cells. So we can see the difference between brown fat and the white fat. The white fat tend to install more newly synthesized fat. And we can quantify that because we can have the linear relationship between the intensity and the concentration of the chemical bond. So we can do quantification. This is the same thing here. The sebaceous glands, we can see layers of cell that synthesize the new lipids in a different time scale. So for 2 days labeling compared with 8 days labeling, we can see the signal starts from the edge of the sebaceous glands. Then they start to migrate towards the center. And after 26 days, we can see the whole sebaceous glands become red, which means those outside the cell have a higher metabolic activity. They tend to synthesize lipids much faster than the cells in the center. In the center, the cells, they sacrifice themselves and secrete the lipids to moisturize your hair and the skin. And for the aging study, we can visualize how C. elegans synthesize new protein, but when they become very old, those newly synthesized protein could not be uptaken by the germline anymore. So this protein were produced specifically for germline development. But when they become old, they are not used or uptaken anymore. So they float around and distributed in the whole body, which is very harmful for the old C. elegans. Sad. Another application I want to mention is we can visualize tumor boundary more clearly. For example, the colon tumor here, if we give a tumor-bearing mouse 3 days of heavy water to drink, they will incorporate the CD-labeled macromolecule in the tumor first, but not the surrounding supporting tissue, the healthy tissue. And in the glioblastoma, the brain tumor is the same. The brain tumor tended to incorporate more CD-labeled molecule inside the tumor cell. So you can see the single tumor cell here, and these are the brain tissue. They have a lower CD signal. So we can draw the boundary between tumor and the normal tissue. Actually, in the hospital, it's really important to recognize the boundary, because the medical doctor or surgeon wants to remove every single tumor cells. They don't want to remove too much brain tissue, but they don't want to leave any tumor cell inside. They will grow into a new tumor very fast. So this will help people to recognize those tumor cells and clean them. To summarize, the heavy water technique do assess the glucose is dried, allow us to monitor metabolism over time and to study many different biological questions. So we also, first time we see the delabeled lipids in vivo, which is nice that we can do time-lapse image. And these two methods can be adapted to a broader range of biological systems and to visualize metabolic activities in subcellular resolution. And we expect that we can use this method to do some clinical application to track the metabolism in human in the future. So I would like to thank my previous mentor, Dr. Wei Ming from Columbia University, and our team members, and my current team members, and funding source, now our collaborators from Columbia Medical Center, especially from, they are from Columbia Medical Center. We did some experiment on C. elegans. So thank you so much for your attention. And I would like to take any questions from you.
Thank you, Dr. Shi, for this great presentation. beautiful metabolic images in diverse organisms. So yeah, please feel free to post your questions on chat, and then we can help Dr. Shi answer. Any questions? In the meantime, I can ask maybe possible question. So these are great metabolic images, and you typically feed some kind of chemicals to the organisms and such, but how do you see the clinical applications of this? Is there any translational pathway, or what is the kind of solution here?
So for the, especially for the heavy water labeling, so we already have some experiment collaboration going on in human body, so that we can let the patient who has the metabolic syndrome or metabolic diseases to drink some heavy water, and we can look at the metabolites in the plasma, the blood samples first to visualize those protein synthesis with CD bond, so we can see those chemical changes from those body fluids first. But we can also do for them the skin side. The skin is on top, so the light can catch those metabolic activity easily, quite easily, for even for skin cancer. So skin cancer cells have a different metabolic activity compared with other skin cells. So we have this advantage to visualize with the spatial distribution.
Exciting. So there are two questions now. Gabriel Lomede is asking, what are the limits to how deep you can image live tissue?
Yes, so people always ask this question. Actually, this depends on the laser wavelength. So we use the same similar wavelength range as the multiphoton fluorescence microscopy, which means we have the same penetration depth as the multiphoton fluorescence microscopy, or two-photon fluorescence microscopy. Yes.
And another question.
Resolution.
Mm-hmm. Another question is from Esra Sengul. Thank you so much for the great talk. I would like to ask for tissue accessibility. The GBM images were amazing. How can you overcome very high infiltration difficulty in viewing it? And do you set any safety threshold in the laser pulse rate for future applications in humans?
Oh, for actually that image from glioblastoma, I took out the brain tissue already. So that is from the mouse xenograft model. For the human glioblastoma image, I think we have to open the skull if the surgeon is doing the surgery. So in that sense, it's possible to use the technique during the surgery, or when the surgeon take a piece out from the edge of the tumor. When they remove the tumor, they have to do the histology. So we could do the histology much faster. Typically, traditional methods will use H&E staining, so they will take a few hours to let the patient under anesthesia for just waiting for the results for a few hours. So now with this technique, we can do it in 5 minutes. So you can visualize under the microscopy just the same piece of tissue that the medical doctor wants to confirm if there are still tumor cells left inside. But it's much faster now. So you can visualize the same histology image. We call it digital histology. So we can take the protein and the lipids channel and to visualize if anything left in the boundary. So that's, I think, the most practical clinical application during surgery for the glioblastoma case. And I don't know if you have any other. Did I cover your question yet? Did my answer covers all the questions from the glioblastoma question?
Yeah, I think probably, yeah, definitely. Yes, definitely. Yeah. Thank you so
All right.
much, she
Thank
said.
you. Thank you.
And as the questions are coming, I have another one. So what is the cost and availability of these machines, right? I mean, it's a microscope as far as I understand, right? And will that be like cost effective that can get into some kind of hospital, or like what is the cost range or availability?
For the cost range, so currently not very commercial available yet. Although some companies, they are pushing forward the commercial to commercialize, like Leica, they are pushing forward the commercialization. So the Leica, their price, perhaps if you want them to provide a commercial available one, maybe around the 1 million or 1 point something like 1 million. However, we built our own microscopy in the lab that like a half price, not half, just like 650K range. So you could buy parts, then you make them you just assemble them together by yourself and configure the signal. But for the hospital, I think the commercial piece will be more convenient. It's easier to carry and operate. So just spend like 1 million, perhaps it's reasonable.
Yeah, of course. So in hospitals.
Yeah.
And yeah, as also questions are coming. So as you know, this is a Spatial Omics seminar series. So is there any plan to make these highly multiplex? I'm also aware of this Nature paper. And what is the maximum number of things that you can look at in a single experiment?
It's a very good question. So it's also our future direction that we could use the clustering technique to separate more molecules, because the Raman spectral information include those chemical, bond signature information. So if we do the clustering technique and to separate those molecules more like in a refined way, we could visualize many more types of real multiplex, like 25 colors, just separate those molecules. And by clustering them, we would see much clearer, like what types of molecule, even the subtypes of lipids, the saturated, unsaturated, phospholipids, or the cholesterol, and some subtypes in the brain. A lot of different types of lipids have their own signature. Triglycerides, they have different Raman signature. So in current status, we only call them lipids, and you can do clustering to have multiple color of subtypes of lipids separated. So it's a multiplex image for many more molecules that we can confirm the signal with other technique, validate them with other technique. So this step is not being done, has not been done yet, but it's very exciting to pursue that direction.
Thanks. And another question is from Julia Velasakis. Thanks for your great talk. Does tissue tumor resection cause stress to the cells that could alter the metabolic signatures? How long are these signatures stable after resection?
Oh, this signal, if they incorporate it into macromolecule, the signal is stable for a long time. So if you fix them with paraformaldehyde, it can stay for very long time, actually, very long time. Yeah, it doesn't go away if it's incorporated into protein or lipids or those macromolecule. But if this signal is in the small molecule, small metabolites like choline, like amino acids, then after fix with paraformaldehyde, these signal will be gone because they are not holding those small metabolites. So it depends where they incorporated.
And maybe one more final question if you guys have it. Okay, if not, then let's thank the speaker again. Thank you so much.
Thank you very much.
And this ends our today's seminar. And next week, we will have Dr. Sinem Saka from Harvard University in the team of Pang Ying. And she's going to be talking about immunosubbur, which is a technique to perform multiplex protein measurements. And I hope to see you all and have a nice weekend.
Thank you.
Thank you.
Thank you so much for the invitation. Thank you for everything. And have a great weekend.
Sure.
Bye.
Bye-bye.
Bye-bye.